Identification of cell death. Standards for the identification of cell death have changed. Cell death used to be defined and described based on morphology. Now there is a switch in classifying it basing on molecular and genetic definitions. This description is more functional and applies to both in vitro and in vivo, so cell death subroutines are now described by a series of precise, measurable biochemical features. A set of recommendations for describing the terminology of cell death was proposed by the Nomenclature Committee on Cell Death in NCCD in 2009, because misusing words and concepts may slow down progress in the area of cell death research. The classic definition of death defines it as a state characterized by the cessation of signs of life. It is when a cell has lost the integrity of its plasma membrane and or has undergone complete disintegration, including its nucleus, and or its fragments have been engulfed by a neighboring cell in vivo. It is caused by an irreversible functional imbalance and collapse of the internal organization of a system. The role of cell death is the maintenance of tissue and organ homeostasis. For example, the regular loss of skin cells or a more active role seen in involuting tissues, like the thymus. Cells die either by accident or design. In fact, there are two mechanisms of cell death, necrosis and apoptosis. Apoptosis in invertebrates is called cell deletion. Dying cells are engaged in a process that is reversible until a first irreversible phase or point of no return is trespassed. Necrosis is an unprogrammed death of cells, which involves early plasma membrane changes leading to loss of calcium and sodium imbalance. This causes acidosis, osmotic shock, clumping of chromatin, and nuclear pycnosis. These changes are accompanied by a loss of oxidative phosphorylation, a drop in ATP production, and a loss of homeostatic capability. There are also mitochondrial changes which include calcium overload and activation of phospholipases leading to membrane diffusion signals, a stage of irreversible damage. The secondary stage involves swelling of the lysosome, dilation of the endoplasmic reticulum, a leakage of enzymes and proteins, and a loss of compartmentalization. Apoptosis, or programmed cell death, is generally characterized by distinct morphological characteristics and energy-dependent biochemical mechanisms. It is considered a vital component of various processes of life, including normal cell turnover, proper development and functioning of the immune system, hormone-dependent atrophy, embryonic development, and chemical-induced cell death. For example, the differentiation of fingers and toes in a developing human embryo occurs because cells between the fingers apoptose, resulting in separate digits. Cell death methodology. The morphometric method. The morphometric method is a way to demonstrate cell death in the laboratory. Morphometric measurement provides the result of cell death as a volume, size, weight, and length of tissue, organ, and the whole organism that compares with before and after the occurrence of cell death. This method was observed by Atala and Johnson, who used electronic particle analyses to determine cell viability. Histochemical and autoradiographical techniques. Another indicator of cell death is acid hydrolysis, which is released from digestion during phagocytosis of dead cells by macrophages or neighboring cells, and the intravital dye is a marker of secondary phagocytosis. Histological and cytochemical techniques. To demonstrate cell death, in some cases a vital dye is used to detect when cellular function is disrupted. This procedure uses living tissue that is immersed in diluted 1,0 solution of Nile blue sulfate in saline. The measurement of cell death by using this dye is observing a change of color or the formation of fluorescence. When the cell died, the nucleus went through destruction stages, one of them pycnosis, which lead to the release of a basic histone group, and this happened when the irreversible condensation of chromatins occurred. The phagocytosis process took place in secondary lysosmes, and the autophagy and heterophagy controlled the dead cell by acid hydrolysis activity. The techniques used to explain this is by the detection of 6-3-H-thymidine and acid phosphates activity in cryostat. Procedure. Specimen was injected with 6-3-H-thymidine, and then the tissue was sacrificed, removed after 1 hour, and quenched in liquid nitrogen. Then 4 cryostat sections were cut and mounted on clean cover slips. The cover slips were held with sections in cryostat, fixed in cold annular acetone for 10 minutes, and the cover slips were rinsed in buffer incubated in acid phosphate medium 15 minutes. Naphthol STR phosphate was used as the substrate and hexazonium paraosinolene as coupler. Again, the sections were rinsed thoroughly in distilled water and the sections were dipped in autoradiography emulsion, ionifert L4 diluted 1,5. Preparations were exposed to B0-4 deg C2 to 3 weeks in darkroom. The photographical slide was processed, counterstained in hematoxylin, and mounted for microscopy. Results. The result of this experiment is the red color, which is caused by ozodye technique done above, and this is an indicator that cell autolyses occurred. This was the major aim in the morphometric method. Another thing is the production of silver grains in the photographic emulsion. Discussion. This change in color is due to fine homogeneous red reaction of acid phosphatase activity. In the lysosome, there is a lot of indication of cell death like the free hydrolase. Incorporate tritiated thymidine gives silver grains in the photographic emulsion which happened in the cell nuclei. The ideal tissue for this procedure is thymus tissue. This discussion focuses on 2 changes that occur in the thymus of mouse as an example study. The first change is the ratio of cells that are dying diffusing acid phosphatase, and the second is the thymidine incorporating cells synthesizing DNA. The results are compared according to the age of the mouse. After measuring the ratios and numbers, the conclusion is that the level of cell death in involuting thymus doubled in comparison to the young thymus, and the thymidine decreased in the older thymus compared to the young thymus. To further show these results, it was observed that some thymocytes contained lysosomal sites of acid phosphatase activity. When the macrophages engulfed the dying cells, the levels of acid phosphatase increased. Autoradiography technique with histological staining. Louis employed autoradiographic incorporation of 3H-thymidine to calculate mitotic indices and estimate pycnotic indices. This technique can be used to study the tissue kinetics of tumors and has applications in the scanning electron microscope. Scanning electron microscopy, ROScopy. The scanning electron microscope was employed by Hodges and Muir 1975 to study autoradiograph. This approach was combined with the cytochemical method for demonstrating free acid phosphate and cell lyses. Dying cells which are rich in free acid phosphate will contain a brominated reaction product and will give a characteristic signal for bromine when subjected to X-ray microanalysis. Fine structural studies. There are common fine structural changes occurring in dying cells. This was concluded after some attempts from the scientists like Kerr 1972, who proposed the general concept of apoptosis in vertebrates, while Schweikel and Merker 1973 described induced and physiological cell death in prenatal mouse tissues. Using fine structural distinctions, it is possible to recognize and differentiate between the types of cell death, acid phosphate, and cell deletion. Acid phosphate is an enzyme or group of isoenzymes which can be used in demonstrating cell death, and this can be easily observed by the electron microscope. Phenitrophenyl phosphate activity can be used as a good marker for cell death. This marker has been used to localize the cell death in cells during embryological development, and the result noticed was the release of exoplasmic non-lysosomal acid phosphate. This appears as a sign of cell death. There are many experiments showing that the ectoplasmic p-nitrophenyl phosphate, which was released, is related to ribosomes, not to lysosmes. DNA, RNA, and protein synthesis and cell death. Programmed cell death means genetic control of the process, thus genes specifying cell death in a developmental sequence must be present. Many authors show that there may be a premonitory increase in protein synthesis as a primer for programmed cell death. References